Well, welcome to the audience, and I want to start by saying that I'm very honored to be a part of the Sakura webinar series for this season, to be on their faculty. I also want to commend all of you who are in the listening audience for taking part in this. Many times, especially if you've been in histology for a number of years, you look at some of the format that's presented, and it's something that you've had for years or you've had years ago. But I always like to say that even taking something like this as a refresher continues your knowledge and your growth and development as a healthcare professional. And moreover, it helps you interact with young technicians that are coming in who have not had the opportunity to possibly go to a school or to have some of the intense training. Many times today, technicians are brought in and taught on the job, which is good, but the intense training that we used to have, I think, oftentimes has been replaced because of healthcare today and the need to just ramp up. So this can actually act as a benefit to you to help train new techs coming in, because they certainly need the advantage of your influence in their lives, in their young development. So what we're going to be talking about today is basic staining of microorganisms and tissues. This is a very basic lecture, and we're going to just talk about some basic things such as morphology, staining techniques, the mechanism of staining, and so forth. And I am technically challenged with my cursor because I can't find my advance. So I'm going to just go ahead and use the arrow. The workshop objectives today are to give a basic understanding of morphology, and also those of you who have the handout is exactly the way this webinar is going to go, so you can follow me along with that. But to give a basic understanding of the morphology of microorganisms, to demonstrate the mechanism of staining to identify the specificity of the microorganisms, to give practical hands-on experience to the basic staining techniques. Now, normally when I've taught this in the past, it has been as a wet workshop. Today, unfortunately, because of the webinar format, we won't have that opportunity to do that. But I have done this at state and national meetings, at the NSH meeting. And so we're going to follow the same format today with the exception of actually doing the stain. And lastly, to present a cost-effective alternative to automation for some of the very simple staining procedures for microorganisms. Many of the labs today are automated, and this whole procedure has been automated. And one of the disadvantages of that is that we put things on a machine and the machine does the work. We sometimes don't understand the theory, the mechanism of staining, and so forth. If something happens within the procedure or on the instrument, then it's hard for us to discern what happened and where the breakdown was. And oftentimes we just repeat the stain. So it's important for you, whether you're using automation or not, to at least understand the theory behind what we're doing. And also, if you're not doing automation, there's very cost-effective methods of doing this for something as simple as a gram stain and so forth. If you're doing a high volume, then yes, automation can be very advantageous to you. But if you're only doing a few slides at a time, there's much more cost-effective ways of doing that, and we're going to discuss that today. So one thing that I like to do when I start is to use this slide, and I ask the audience to identify the stain. And so also in identifying the stain, I want you to look and just give some thought as to how you would grade the stain. What entity is staining specifically in this stain? And when I do the workshop, I have them write this down as how they would grade this. But for the sake of today, just write alongside on your handouts on a scale from 0 to 10 how you would grade the stain, 10 being the highest quality. You might even want to note what type of stain you think this is, and also what entity is staining in this. Now I just advanced to the next slide where you can see some arrows now. And so actually the entity that is staining within this stain are highlighted by these arrows, but for certain reasons you can identify them readily. And so we're going to talk about that a little bit later, But one of the main concerns that a histologist would have if they're looking at this is that the background staining is so dark, so heavy, that it's masking out whatever staining entities that are there. Thus, the explanation for microorganisms, because they're so small and so minute that it is very easy to mask out the specific organisms by the counterstain, or if the stain is not done optimally. And if you cannot identify them, then you cannot look at the morphology and you cannot make an exact assessment of specifically what type of microorganism this is. So the next slide basically just talks about what are microorganisms. They're small life forms seen in systemic infections that actually, the nature of systemic infections is that they spread throughout the body. They grow basically as a colony, and then by some form or another, they will spread throughout the body and cause major medical problems. Traditionally, they're small, so small that they require microscopic observation. And because of this, there's a high risk of false negatives because they're so small, and they have to be observed microscopically. So far as medical reasons, they're divided into 4 primary groups. Microorganisms are bacteria, fungi, viruses, and protozoa. And for the sake of this lecture being a basic lecture, we're going to basically talk about bacteria and fungi. Now, some more information about microorganisms is that they are unicellular. The bacteria, we'll just talk about that first. They have genetic material within the organism, but it's not enclosed by a distinct nuclear membrane. So that's very interesting in how they produce. Each individual cell has the ability to metabolize and grow and reproduce, which actually adds to the systemic nature of the infection. They can rapidly grow, they grow in colonies, and they can reproduce readily, and they can spread readily throughout the body. The classification primarily is based on the morphology of the bacteria, whether it's oval or spherical or spiral. That's basically how they classify each bacteria. Similar to plants that have a cell wall, they do have a cell wall, but without chlorophyll, and they are colorless. So actually when we're staining, some of the things that we have to be cognizant of is the colorless cell wall. So the staining methods that we use, we can't rely on being able to color this cell wall, as in many plants or fungi. The cell wall is composed of a form of polysaccharide called peptidoglycans. And this cell wall can, we're going to talk about this a little bit later, the nature of the thickness of the cell wall will also have a lot to do with how these bacteria are classified. They're basically classified in 4 different categories: gram-positive, gram-negative, as differential staining methods, that's 1. Acid-fast, that's 2. Silver methods, that's 3. And fluorescent methods. So these are the 4 different classifications of staining that are used when we talk about bacteria. Now, just giving you a brief overview of bacteria, let's move on to fungi. Fungi, as opposed to bacteria, they are primitive plants in their classification, and they are both uni and multicellular in nature when you look at them morphologically. They have a common membrane, and it is bound enclosing the genetic material, unlike bacteria. So the cell membrane in true plants and true fungi are made of chitin. But there is also a very high polysaccharide layer in this cell membrane. So the groups are classified, the fungi are classified into 4 major classifications. You have filamentous fungi, which are, the morphology of those is you see hyphae separated by septae, which I'll show you a picture of that in a minute. But they also grow in anastomosis, which they branch off as in like a tree branch. Another group is called the yeast, which are oval in morphology, and they were produced by a budding phenomenon. And I'll discuss the budding phenomenon, very interesting, in a few minutes. And then you have a yeast-like fungi, which has a lot of the properties of yeast, but they grow in elongated budding, which is called pseudohyphae. And then you have the dimorphic fungi. The growth and morphology are very dependent upon the temperature and incubation. They can grow as one type of organism, and then if the temperature is altered, they can change and grow in the morphology of a different type of organism. So the last two classifications that I want to talk about so far as microorganisms are the protozoans and the viruses. The protozoans are unicellular, and their main distinctive characteristic is that they have a locomotive characteristic. Some of you all might remember in your basic biology how amoeba have what's called a pseudopod, where there will be a protrudence in the cell wall, and the cellular material will flow into that, and actually the organism will spread into that and move in that locomotive capacity. So it'll branch off in a small foot that comes out of, called a pseudopod, and then the material within the cell will flow into that area, and actually the cell will move in that fashion. Also, some protozoans have flagellae or cilia, which are small projections out of the cell that move and orient the cell that way. Flagellae stick a long projection out, and they will pull themselves through that, whereas cilia have small hair-like structures, and through their movement, they can actually orient and move the cells in a certain direction. The last classification is viruses. They are intranuclear, and viruses are very important because in histology, we're starting to see labs go more and more into DNA and RNA extractions or into molecular biology. The key thing about the DNA and RNA is the protein. There's a protein layer that coats the DNA and RNA that must be lysed to expose the genetic material and get the information that the researcher is looking for. So that is a specific method that has to be used, unlike what's used in the other four classifications of microorganisms. So I don't want to spend too much time on protozoans and viruses, so I'll move on to what we were talking about. Let's go back to bacteria with the basic morphology. And this is the key thing so far as classification and actually staining, the specific staining procedure that you would select. The groups are determined by shape, as I said before, and there are three basic shapes: the oval or spherical. And if you look in the lower, I'm going to put the slide here, this would be the oval or spherical. And then they're also rod-shaped, and then they're also the spiral or corkscrew shape. And specific microorganisms, or excuse me, specific bacteria will have one of these three classifications, and therefore, based on their morphology, they are classified thus. They do have a cell wall exterior, which you can see here. And within that cell wall, there is a colorless lipid layer. And this is another thing that is very important in the classification of bacteria, because within some bacteria, this cell wall is very thick, this lipid layer, which tends to resist certain types of stains. So there are certain types of stains for bacteria that you cannot do on this. And so that's the primary classification, this lipid layer. We're going to discuss this quite a bit in the lecture. So there are basically four different types of staining techniques when you talk about bacteria. There's differential staining, where you would do a stain and then you would actually decolorize it and do another stain over that. Then there is acid-fast stains, which this lipid layer makes it resistant to acid decolorization. The third category is silver stains. And in silver stains, the organism is generally so small that this lipid layer would be stripped out during the procedure, so you would want to use some other kind of method of staining, which is like silver impregnation. So that would be the third. And then the last would be fluorescent staining, where we would actually use a fluorescent dye to mark the bacteria, and then under a fluorescent scope you could see it fluoresce under a dark background. So speaking first about the differential methods of staining, There's 4 classifications that I gave you here, 4 different staining methods that are the most common ones in histology: the Graham method, the Brown and Brin, the Brown and Hops, and the Lilly Tort. And all 4 of these follow one basic mechanism of staining, which we're going to discuss a few slides from now. But here on the right, you see an example of gram-positive staining. And these are cocci, the form of one of the forms of the bacteria. And one way that you can tell differential staining between gram-positive and gram-negative, most characteristically, is that gram-positive will be in a cocci appearance, and gram-negative will be in a rod-shaped appearance. This is specifically Staphylococcus aureus. And in the next slide, I'm going to give you a little bit of background on this slide. Photograph of spherical cocci, gram-positive, Staphylococcus aureus, normally occurring in nasal passages. This can also be seen in skin conditions, sometimes serious if it's because of its systemic nature. It can actually lead into blood infections and pneumonia. So once again, you can see the blue staining, and I'll give you the mechanisms of staining in a minute, but this is the crystal violet picking up on the cocci, the gram-positive cocci. Next slide is also differential demonstration of staining. These are also gram-positive stains, cocci, and this is staphylococcal pneumonia. So just some background on this. This is a sputum showing the Staphylococcus bacteria. The gram staining technique was used in this. What we're looking at is gram-positive organisms in the blue cocci form. This can also lead to pneumonia or other systemic infections. Now, this is one of the common diseases that we often see patients acquire that have suppressed immune deficiency for whatever reason. They're weaker in the hospital. They have other kind of symptoms that their body is trying to fight, and their immune system is somewhat suppressed, and so pneumonia can set in. And this is actually the organism that we see in those patients. So the next slide shows you the gram-negative organisms, and these would be more in the rod-shaped organisms. You can see here that they have the opposite color. They've picked up the red staining, which is part of the differential staining method. You have one stain that you do, and then you differentiate, and then you have another stain that you do. And the first stain is typically crystal violet, the blue, and the second stain would be a counter stain, such as safranin. And here you see the safranin staining, but you do not see the crystal violet still staining here. And so this is the differential method of the differential stain, where you can differentiate gram-positive from gram-negative basically by which organism is going to retain the dye in the stain. So to explain the mechanism of staining, here we have gram-positive and gram-negative organisms, and they all follow the same method of staining. There is a fixation procedure, then there is a staining time in crystal violet, which will basically stain both organisms, gram-positive and gram-negative. Now, then there's an iodine treatment, which acts as a dye lake formation, which helps lock in the crystal violet. But then there is a discoloration method, and you see here the differentiation, whereas the crystal violet in the gram-negative organisms has been bleached out. And the reason for that is gram-positive cells, these, have a much thicker cell wall and composed of lipoprotein and form of polysaccharide. This makes the cell wall a lot more permeable and allows it to lock in, and it also allows it to be more resistant to decolorization, this thicker cell wall. The gram-negative, having a thinner cell wall, can easily be decolorized by whatever acid solution that you're using, so it'll strip out that crystal violet. Then the final step is a counterstain of safranin. Now, because of the dye lake that's been formed with the crystal violet, the safranin will not overwhelm or stain over the crystal violet. Because this has been bleached out, the safranin, the gram-negative organisms, are readily available to take up the safranin and stain that. So thus you have your differentiation between gram-positive and gram-negative organisms. And this is very important to clinicians to distinctly decide what type of bacteria this is and provide the adequate form of therapy. So in the next slide, you see just a slide that I've taken just to show from a website, just to show gram-positive organisms and gram-negative organisms. And here are some of the names of the very common ones that we see on the top, and some of the very common ones of gram-negative that we see on the bottom. This one, Escherichia coli, is very common. This is one of the main contributors of some of the health issues that we have in the food industry today, where you have seen on the industry or the news that certain meat products or certain produce products have been infected with Escherichia coli. This is the bacteria that they're talking about with that. Let's talk about the staining mechanisms for, we've talked about the staining mechanisms for, staining types for differential diagnosis, which is gram-positive, gram-negative. Let's talk about some of the non-differential ones. Now, one of the first things is that you have the acid-fast organisms that resist acid decolorization, because one of the key things is it has a much thicker cell wall. Remember I talked before about the thickness of that cell wall. The lipid layer in that cell wall is much thicker and causes the organisms to resist acid decolorization. Because of the thickness of the cell wall, and this is very key, there is a much slower penetration of the dye, whatever dye that you're going to use. Usually it's a carbofuchsin type of dye or something with a phenol nature. But because of that lipid layer, it takes a while for that to penetrate. In the former methods that we used to have, the older procedures, the room temperature procedures, it was somewhere around 30 to 45 minutes sitting in the primary dye solution, the carbofuchsin. And that's because of the slower intake because of this lipid layer. Now with microwave methods and so forth, the heat can enhance the penetration or speed up the penetration, of that dye into the cell wall. So that's one reason why it is very advantageous to use mild heat or either a microwave method when we're doing acid-fast dyes to speed up the time and enhance the uptake of the dye into the organisms. The other method is silver impregnation. Well, first of all, some of the key methods of acid-fast are Kenyon's, Thiel-Nielsen's, and Phyte or Phyte-Furaco. These are the most common ones that we see used in acid-fast. Then there's the silver impregnation, where typically the organisms are so small that when you would use the decolorization step that you would use in acid-fast technique, it would actually strip the dye right back out. So with those organisms, you want to use a silver impregnation technique, which actually takes up silver into the organism and allows it to be visualized in the morphology a lot easier. Some of the key methods are Steiner, and then there is also a Steiner-Steiner technique, Wolf-Starry, and the Dietl technique. Typically, the Steiner is more simplistic, and it is easier to avoid background staining or overstaining. And so, unless there's a specific reason for using Wolf-Starry or Dietl because of the nature of that specific organism, Steiner is usually a more preferred method in routine histology. And then finally, fluorescent technique, where you use a auramine or rhodamine dye complex, which actually colors the organisms with a fluorescent dye. And then, unfortunately, with this, you have to use fluorescent scope. But it's very strong visual nature when you look at this because if done correctly, you will have a very dark screen, and these things will light up so bright that it's almost impossible to miss them. So those are some of the primary staining techniques for non-differential staining. Now, let's go back to bacteria and look at the acid-fast techniques that I mentioned. The primary techniques are Kenyon's, Thiel-Nielsen's, Phyte, and auramine rhodamine. Now, the thing about this is that these carbofuchsin-based or phenol-based dyes in these three will actually penetrate very slowly, as I mentioned before. With one of the procedures, the fight method, and we see this in the earlier methods of the Lee Luna manual that was produced by the AFIP in the early 70s. This method, the fight method, requires the use of peanut oil and xylene mixture before you start the staining process. Now, the reason for this is that, as I mentioned before, some of the organisms are very large, and they have a huge lipid layer. But some of the other organisms, such as leprosy, well, some of the larger organisms would be like tuberculosis. Huge lipid layer, very easily to do with these type of staining techniques. Now, with the fight method, that's more designed for things like leprosy, which is a much thinner organism, which has a much thinner lipid layer, which can easily be decolorized out. So in this method, it's very interesting that you would use a xylene and peanut oil mixture to actually deparaffinize the slide. The theory behind that is the peanut oil would help enhance or coat that lipid layer and thus make it more resistant to the decolorization technique once you go to your primary dyes of your carbofusion or your phenol base. So that's one of the key things, to maintain or increase that lipid layer. And the lipid layer actually gives it the term acid fast, because it holds on or resists the acid nature of the decolorization. So the next method that we talked about before are the spirochetes, and these are some of the 5 methods that are used: Steiner, Wathenstory, Dieterle, Pfeiffer-Rocco, and Cherukhian modification of the Steiner technique. And so we see this in much thinner organisms, such as the spirochetes that are used here, that are seen here. I'll give you show the slide here. Let me back up. As you see here, this is an example of Treponema palladium, the organisms that are characteristic in syphilis. This is a section of testes, and you can see in this organism, on this slide, the typical corkscrew-shaped structure or morphology of this type of organism. Unlike the tuberculosis, which is basically oval, you see this corkscrew-shaped appearance. And I really like using this as a teaching slide because this is perfect to actually show the morphology of this type of organism. Now, this organism is much thinner, and this is another reason why this organism would not be able to visualize this in the typical methods that we talked about before with acid-fast techniques, or certainly with the differential techniques that we mentioned. So once again, the very corkscrew-shaped nature of this, and this is the syphilis organism. We can also look at another organism that is more advantageous to use a silver technique, and that is what we're looking at here, which is seen under gastric biopsies, and this is H. pylori. And they're very difficult to see because they look like small cigar-shaped organisms, and typically these are seen in the crypts or the glands of the intestine, and this is one of the main contributing factors to the appearance of ulcers. These organisms grow, they typically eat away the stomach lining, and ulcerations are formed. Let's move on to the mechanism of staining that we talked about for the silver techniques, or actually let's go back for the acid-fast techniques. The mechanism of staining is, as I mentioned before, a phenolmethane dye in a phenol solution. Usually this is a carbofusion type of dye. It's a slow penetration, as I mentioned before, so you have to allow time, or you have to use heat to enhance the penetration rate. Then there's a decoloration step, usually with a mild acid alcohol, and then there's a counter stain. And now the significant thing about the counter stain, as you can see here in the picture, is that you want a counter stain that just adds a light contrast to your organisms. Now this counter stain here, this is the Mycobacterium tuberculosis, and this is an acid-fast stain. And the tuberculosis bacilli are staining red. And even in this, the background staining of this is almost too dark, because you really want these organisms, because of their very small, minute natures, to jump out at you. So I would suggest on this staining procedure here that this would be the absolute most that you would use on your background stain, if not less. Now this takes us back to the first slides that we looked at. As you can see here, these are the slides, and this is a very good example of overstaining. I put these arrows here to show you actually where the organisms are, but because of the nature of this counterstain, it's impossible to discern these individual organisms, and it's really impossible to look at the morphology and validate that they are specifically tuberculosis bacilli. Whereas in this one, you can see a much lighter counterstain. You can see the organisms staining, and once again, one of the good things about this is that they stain in colonies, or they grow in colonies, so it's easier to identify them as patches. Once again, this counterstain can be a little bit lighter also. I'm going to switch to another example, which is the same here. Same example up here, counterstain much too dark, and here more optimally stained where you can see the colonies. Now in these slides, you would actually want to look at them under oil immersion optimally, because that would then just switch to another slide, the last example here. You can kind of see morphology, but you can't see color or anything like that. And in a true acid-fast stain, the acid-fast bacteria are the only things that are going to retain the red dye, the phenylalanine dye, which here you can't really tell that. You can just tell morphologically that there's some type of structures there. Here you can actually see them retaining that dye. So moving on to these silver stains that I mentioned before, I go back to the class and I ask them to grade this stain. And so I'll ask you all that too. And if you can note how would you grade this stain, and if there were any issues with it, what would you say are the issues with this stain? Some people have said this is an optimal stain because you can see all the organisms staining, which is a valid point. But when we look at this, remember, we want to make sure that we can specifically identify the exact organisms that are staining and not some other entities. So in this next slide that I'm going to show you, I also ask the class to identify these organisms all in here and rate the quality of the stain. So once again, on the last slide and on this slide, from 0 to 10, you can actually put down just what your assessment is. And we're going to discuss this a little bit later also. So silver stains are primarily used for, well, one is for the medical groups of fungi. And if we look back at fungi, we'll go back over that. From a medical standpoint, fungi are basically broken down into 4 different classifications. I mentioned this before: filamentous fungi, yeast-like, and dimorphic fungi. There's basically 2 different classes: the superficial or systemic fungi. Superficial would be something that you would see growing like on the skin as a skin lesion. Sometimes you see athlete's foot or whatever type of lesion, but it's basically superficial and it's localized, as opposed to systemic, where you see it spreading throughout the body. And this is also how they classify different types of organisms. The morphology, or filamentous, phi-hyphae, or oval. So when we look at fungi, they're going to be in one of these 2 morphological classifications, and you can see how they grow. One will be by budding, and one will be by an exaggerated type of budding. And I'm going to explain all of that in just a second. Well, as I mentioned before, I don't think the budding phenomena, let's talk about that for a minute. Budding is a very interesting phenomenon that forms. And what you'll see is primarily on organisms that have this oval morphology, you will see an extension formed on the outer surface of one of the organisms of the parent cell. And the nucleus of that parent cell will divide, and one of the nuclei will migrate to the bud section. And then the cell wall will come down, and actually the bud will separate and break away. So this is the method of reproduction that we see in budding. And so when we look at the morphology of fungi, we will see that phenomenon here. Right now I want to show you, before we actually look at that, I want to show you the mechanism of staining for silver techniques, since that's what we were talking about. Basically, this is the GMS procedure that most histologists are familiar with, Grocot's methenamine silver. And what we're going to be looking at here is Aspergillus fumigatus. So the first method is generally an oxidation of the tissue and the fungal polysaccharides, typically by chromic acid. Then there is a reduction or rinse to remove the trace oxidant, and then it is exposed to an alkaline silver solution, typically methanamine, which lays down the silver layer or silver impregnation. Then there is a toning procedure, usually used with gold chloride. And what this does is this changes the nature of the methanamine, which is by nature a type of brown metallic, into a black or gray type of metallic. Then the excess silver is reduced from the tissue, and then a very light, once again, light counterstain is applied. So the next slide will actually show you, as it loads up, an example of Aspergillus. Now, what we mentioned before was it grows in this anastomosing type of morphology, where you will have one organism, it will grow by a false type of budding into another organism, and then that will grow into another organism. And what you see here is this anastomosing, or this branching, or of fibers from one to another. Typically, they stay in long, elongated organism structures, which are basically linked, but they're all individual structures. As we move on to the next slide, you can see a closer shot. And one of the things about this is that with the silver impregnation, this will continue to develop more and more silver to the point of developing background. So you don't want this to the point that you can't see the translucency between the hyphae. These hyphae form. These hyphae are separated by these septi, these separation points here, which this grows into this, and that grows into that. And what you see here is very interesting. Because of the anastomosing nature, what we're looking at here is these grow in all different directions. So you see this growing in a certain direction, and then all of a sudden you see this here that looks like an oval shape. Well, that is actually the organism now growing towards you. And so as we take this section in microtomy, we're looking at a cross-section of it. So even though some of these look like buds, they're still actually the anastomosing elongated fibers. Another thing to note in this is the light, delicate nature of the background stain, the counter stain, which is a light green. So optimal staining is that you have to have the effect of the silver impregnation only to a certain point, so that it doesn't avoid you being able to see the translucency. You should be able to see a light gray appearance, not black, and then just a light laying down of the counter stain. Now another example here that we see there. So now another thing to see here is you see a variability in the nature of the stain, in the intensity of the stain. And that's one key thing, because these organisms will not all pick up silver at the same rate. They will primarily at the same rate, but this is why this stain has to be monitored, because you see here very distinct septi separating the organisms. You see the darker gray appearance here, very distinct, but you see a lighter gray appearance here, darker here. So traditionally, in the older days, we would have to monitor this under the microscope. We'd have to go back and forth in the staining in the silver solution, and then rinse very quickly, put it under the microscope, and then go back to the silver solution. A lot of the procedures today, most of the procedures today don't require that, but optimally you would still want to do that. But it does require close monitoring, whether you go back and forth to the microscope or not, because it doesn't take long before some of these organisms are overstained, and they pick up so much silver that you cannot see the hyphae in between these. And therefore it makes it difficult to distinctly identify the morphology and identify specifically what type of organism this is. It's very easy to misdiagnose these organisms. So this takes us back to, this takes us on to another type of microorganism, And this organism here, you can see is much different from the Aspergillus. And this is pneumocystis. And this is the opportunistic infection that we mainly see in HIV patients. But anyone basically who has developed pneumonia, this is the type of organism that you see basically in their lungs most often. This is a section of sputum. And we can see here with the pneumocystis, they have a small kind of oval shape. It's almost like a helmet type of appearance. And they have this small little belly button type of structure called connectopore. So once again, it's very difficult to distinguish some of these organisms because they turn at different rates. So with some of these, you can see them turning very lightly here as opposed to here. But some of them are more optimally stained. And that's why the nature of this stain, just like the other silver stain, is that it has to be monitored very closely. So this takes us back to some of the other slides that I showed you before. Now, I asked the question before about how would you rate this stain. So we see a lot of things in here picking up the silver impregnation, but that doesn't necessarily mean that all of these organisms or microorganisms, red blood cells will pick up silver, and they will darken. And so these more than likely are red blood cells in this slide. But you can still see the Aspergillus, the anastomosing hyphae, here, stained very light gray, as opposed to these other organisms. So this is one of the main reasons why you have to have optimal staining, and you have to have good visual morphology, so you can determine. Oftentimes this has been misdiagnosed because that lab, they just saw this as, oh, it picked up, so they thought that this was part of the staining, and it wasn't. And so like the next slide that I'm going to show you, these are, this is one of the slides that you graded before. These also are organisms mixed in with some other type of organism, probably red blood cells. But you can see the nature of these. This might be blastomyces or candida. And I believe this is candida. But you see the oval appearance. But you see them a lot differently from these other oval-shaped appearances. And so more than likely, and these are red blood cells that are picking up, as opposed to the true organisms that are picking up here. Once again, this calls for the need to have really good quality control when you do this and to be able to monitor it very closely. Now, the silver stains is one of the things that actually led me to believe that automation can be a very viable asset to a laboratory in special stains. Years ago, when they first came up with special stain instrumentation, I was very reluctant to use them because of the nature of silver stains. Silver stains have to be monitored very closely. They have to be cut off at a certain time. And so that was very difficult, even from a manual perspective. But I realized later on with the instrumentation that came out years ago that they were able to achieve this optimal staining. And so I think that there is something definitely to be said on the positive for automation when it comes to silver staining and any other staining technique. Now, you can be using automation for these, or there are other cost-effective methods of doing these stains. If you're a laboratory that is only doing like 1 or 2 stains a week, very seldom, it doesn't really, it's not really cost-effective for you to invest in huge automation, even with the expense of the instrument, the expense of the reagents and the kits, and you're not running that much. These are some of the companies that actually have commercial vendor kits that are on the market that are very easy to use, very common in many laboratories. I'm not really recommending one over the other, but they are all very good companies to use, and they all have very good procedures to use. So I would recommend, if you're a smaller environment, to try using one of these methods. But certainly, once you these kits are very good for you to be able to learn what you're doing, even if you are using automation. So doing the step-by-step procedure will give you a better understanding of each step, as we talked about in the mechanism of staining. So this lecture today has been pretty brief, and I welcome any of you all to look on the NSH schedule, those of you who might be attending next year, or this year in 2014. I have submitted this to be taught as a wet workshop, and it is very informative to follow up the theory that we've just talked about with actually hands-on, because you can actually look under the microscope and see the development of the stain in each step. So those of you who might be interested, when the program comes out, you might see if this is one of the ones that are on the schedule. I actually did this for the last 2 years in NSH, and it was received very well. Some of the references that we used today for this lecture, as you can see, the Carson, Bancroft, Sheehan, some of the classic ones, and some of the websites that I use for the images. So once again, the purpose of this lecture today was to give you basic understanding about microorganisms and their identification, their morphology, and their staining techniques, how one differentiates from another, one microorganism differentiates from another, and how important it is to be able to do the stain optimally so that the pathologist can visually validate the type of organisms that he thinks it is. Many times the pathologist can look at an H&E and they can see tuberculosis bacilli in there, but the confirmation of that comes when they do the special stain. And the pathologist will rely on the technologist to give him the special stain that will actually visualize that organism and nothing else. So once again, I want to commend you all for your attendance today. I want to thank Sakura Finetech for actually putting on this webinar series, and I want to commend them for being the type of company that provides continuing education in the industry rather than just being a company that sells a product. They are investing in the growth and development of the technologists in the industry. And with that, I want to turn the webinar series back over to Alicia Rios, who is our coordinator. Thank you.
Thank you so much, Skip. That was such a well-informed presentation with a lot of great examples. I would like to take this time now to offer up any questions. If the audience has taken any questions during the webinar, please type those into the question box. And while you're doing so, I would like to talk about our featured product of the month. We feel that the Prisma stainer is a complement instrument for this presentation because once it is staining, but it also has special staining capabilities. Many of you might not know that you can use the Prisma stainer for special stains, and there are special reservoirs for doing so. It is a very stable and instrument that is a workhorse within the labs. I'm sure many of you know this already. You might be using it currently in your lab. But it can run multiple stain protocols like histology and cytology, including PAPs and non-gens. If you need more information on this product, please reach out to your local Sakura rep, and they'll be happy to share information with you. At this time, I would like to ask Skip some of the questions that came through. Skip, are you ready?
Yes.
Okay. It says, is staining of RBCs an error? Is there a way to prevent RBCs from taking up the silver?
The staining of RBCs is an error because that's not the entity that you're looking for typically when you're staining for microorganisms. In silver stains, it is just inherent that the properties of red blood cells can pick up some of the silver impregnation. When we talked about the chromic acid oxidizer in the beginning of the staining mechanism of the GMS stain, the silver stain, it's such a strong oxidizer, but that it will also unlock some of the staining properties on other tissue elements, red blood cells, because of their properties being one. So it's inherent within the red blood cells that if you have a section of tissue that is loaded with red blood cells, it is important that you know the morphology of those red blood cells as opposed to the organism that you're staining. Now, typically with the red blood cells, they're going to pick up these staining properties a lot faster, and a lot stronger than your organism. So that's one way to be able to differentiate one from the other. Secondly, if you do an optimal stain for certain type of fungi, you will see a staining differential. You'll see the red blood cell staining, as I showed you in the image, really dark, all during the time that the organism is picking up the silver impregnation, which will be a light gray. So it's easy to really visualize one from the other. I don't know of any method of actually avoiding the red blood cells from actually picking up the stain. There may be some, but I'm not aware of any. It's just something that you have to be cognizant of and aware of through the morphology. So that's why one of the things that we always stress is that the histotech visualizes the special stain before they take it out to the pathologist. And I would recommend that if you see the situation, that you put some type of notation to the pathologist that there is a predominance of red blood cells in here and you see some endogenous staining or pickup of silver from that. That way the pathologist knows that you're aware of this, that you're not trying to identify these as the organisms that you're saying stain positively in the slide. So I hope that answer helps.
Thank you. The next question is, we constantly get a fine precipitate on our gram stain. What can we do to avoid this? We have tried filtering and over differentiation.
This is on the gram stain?
Yes.
Filtering is the only method that I knew of that. I don't know if, and I'm saying this just off the top of my head, because filtration has basically taken care of that for me when I had those issues. You might get to the point, I don't know if you're filtering everything or just the gram positive or the crystal violet. Certainly the crystal violet has to be filtered. I think if also the time that someone mentioned to me once, the time that you spend in your fixation can also vary that if the fixation time is not adequate. Now, I never tested that method. That was just something that I heard in conference with another technologist. But I would say possibly your filtration methods, maybe you need to do double filtration. When I've had background or some kind of sediment or some kind of pickup, it's generally been taken care of for me by the filtration method. So I'm sorry that I can't be more direct about that or more distinct about that, but that's basically how I had success in solving that procedure. You have my email address at the front slide, and what I would like to do is get some information on that and communicate back and forth with you with what I've found, because that's a very interesting question. So feel free to email me that question, and I will do some research on that and get back to you.
Thank you. That is the end of our time for today. Look forward to speaking to you all next month on the 27th. Thank you.